Welcome everyone. In my talk today, I'm going to present IFI's brown trout genetic program and some of our more recent research in this area. In the talk, I'm going to look at why we're doing it, how we're doing it, what exactly we are doing, the questions we are trying to answer, where we have done our genetic program so far. I'm going to look at a few case studies, a carbon lock shield, and sorry, I should have updated this one. I'm not going to do the lithium one at this stage. And then I'm going to present some findings, general findings from all the studies we've done so far. So when we look at why we're doing it, there's a whole suite of questions you could ask about genetics. But I suppose really if I start at the kind of high end and we say that the purpose of the brown trout genetic studies that we do is to know and understand what the Irish brown trout genetic structure is like and to know how diverse it is. That is, how many different genetic strains of brown trout we have nationally and also within our catchments as well. Because it's only once we know what we have that we can protect what we have. So we need to understand what we have at many levels, national and catchment-wise. Then again, if I look again a bit more at why we're doing it, well, we've known for a long time that there are subspecies or different strains of trout. The work Professor Andy Ferguson did on Loch Melvin in the 80s and the 90s showed that we had Fergus trout, Gillaroo trout, and Sonhin, and his genetic work showed that they were genetically distinct. We also had the genetically distinct Cronine trout, which are found in the Camp Core, which is a tributary of the Loch Derg system. And you also have the Donnell from Loch Nay. So, but if you look at the trout on this slide, and these pictures were all taken from the late Professor Martin O'Grady, who obviously had a huge interest in brown trout and genetics. But he had this collection of photographs of trout, and we started, well, we always knew it, but when you see them photographs there, it just becomes more obvious that the coloration is so different amongst these trout, and their spot patterns are very different. And these trout came from different catchments, so we asked the question, is their coloration and spot pattern different because of their environment, or is it their genetics, or perhaps it's the two are interlinked? We weren't 100% sure, are these genetically distinct strains or not? So again, we had these list of questions that were kind of going on in our heads that we could make assumptions about, but we didn't have the scientific evidence for it. So again, when we look at why we're investigating genetics of brown trout, the questions are endless. But we started with some of these ones here, in particular natural barriers. These would usually be waterfalls. We have a large number of catchments, and in those catchments we have these waterfalls that are complete barriers to salmon and trout. But we know we have trout upstream of the waterfalls and we have trout downstream. And we didn't know for sure, again, whether the genetics was different between them and how unique the upstream section or trout were. We assume because these waterfalls are there thousands of years that they probably are genetically distinct, but it would be lovely to have that evidence there. And then you have a whole suite of anthropogenic pressures. And a lot of these ones on this list here have been in place for 100 to even 200 years. So that's several generations of trout at which we could have seen a change in their genetic structure. We have pressures like man-made barriers, which includes weirs, culverts, power stations and dams, our large arterial drainage schemes, the introduction of alien species such as roach and dace, but also flora and other fauna invertebrates. And we don't know exactly has the interactions with trout changed or is about to change their genetics. In Ireland, we've had a very intensive hatchery stocking history. Some of our large catchments have been stocked out from the 60s and 70s and the 80s. This has changed more recently. But what impact did this have? Farm trout at the time were all diploid, so there was a potential there for them to interbreed. Then you've also had huge growth, urban growth, and the associated discharges, farming activities, and agricultural runoff. These are all possibly going to impact on the water quality, and then also water abstraction. So we know trout are very adaptive and they will change in their environment, but we wondered, as the trout are changing or adapting to these environmental pressures, are we going to see a change in their genetic makeup? So again, these are all the kind of things that were going on in our minds. And as I said, the list of questions you're trying to answer can be nearly endless at times. I'm going to just briefly describe how we do it. So we'll start really at the very top and say that all of our brown trout genetic projects are completed through partnerships. IFI doesn't really have the resources to do our genetics program completely themselves. In particular, we don't have the laboratory and some of the resources to collect some of the samples. So we do them with a number of partnerships, which include universities. And with that, we've done a lot of projects with University College Dublin, and our more recent ones have been with Queen's University Belfast. They do all our laboratory work, they do genetic data and analysis, and they do reports. Then we also have worked with a lot of our angling clubs and angling communities. They will have collected adult samples for us. That's quite intensive, and again, resources available to us and time constraints. It was very, very useful that our angling clubs came on board with us. They provided a lot of catchment background information and some historical information on the fisheries. And in many cases, they also provided funding along with a number of other organisations. And then ourselves, we would collect the juvenile samples and we provide fishery data and we analyse that and include it in our reports. So it's a combination of the three different stakeholders that provide these projects. Again, when I look a bit more on how we do it, this is kind of getting down to the kind of basic sampling level, getting out there. Once we've identified the catchment we're going to take on next, and we know the questions or the objectives of that study, we then will collect a baseline juvenile sample from around the catchment area, because this will allow us to identify different populations present. Then we'll also collect adults from, depending on our catchment, maybe the large lake system, or if there's no lake, we'll take the large main river. And then we'll use our juvenile baseline populations to determine the contributions each of those makes to the adult population. So we can see if a particular tributary system is contributing a large amount and which ones are contributing a little less. Again, that leads me in then to the sampling methodology. So for our juveniles, we have a catchment-wide sampling for our juvenile baseline population. You really need to get out and sample as much as you can. We're using electrofishing to collect these samples. In our more recent projects, we have started to use trout scales for, well, we don't do the DNA extraction, the laboratories or Queen's usually do it, and the genetic analysis. Some of our earlier projects, we did take tissue samples or fin clips, but trout scales have been found to be quite good for DNA extraction, and it's a non-destructive method of taking our genetic samples. So depending on the catchment, while in theory it would be brilliant to be able to sample every tributary, some of our catchments that we've done this work on have been quite large, such as Loch Carb or Loch Ree, and it, you know, it just resource-wise we weren't able to sample every tributary. And in hindsight, you know, we can see now that we've summed the results back that it would have been better for us if we had. But to continue with the projects, we then would have focused on some of the more well-known spawning and nursery tributaries. And then within those tributaries, we would sample 3 locations and collect up to 50 samples where possible. Believe me, some places it wasn't possible, some of the very, very small tributaries. And then the fish we sampled from were usually 1-year-olds. We were quite, we really needed to make sure we weren't taking very young fish, because that could represent maybe just 1 or 2 families. But by having a number of locations and by taking 1-year-old fish, it meant that we were increasing the probability of having several families, which provides a better profile of the genetic structure.
Okay.
Folks, our adult sampling then, depending on our catchments, we would concentrate on either adults from a main channel, in the case of the Boyne or the Mitigated Shore, and we would sample adults in the lakes, in the case of carp, Ree and sheep, etc. Or we apply a mixed sampling methods because we use angling by our angling communities in quite a lot of our projects. We also use the opportunity to take samples from our own gill netting surveys and small number of places we have some electric fishing in our larger again, we use trout scales for genetic analysis in our adult samples. And but look at what we are doing, what are the questions we're trying to answer. We had, in all of our studies so far, we've had a number of common objectives, and these were to describe the current brown trout population genetic structure of that system and its main tributaries, to quantify the relative contribution of the identified baseline populations to the adult stock. We're also looking to assess the impact of barriers, both natural and man-made, fish migration, and potential impact that could have on the current population genetic structure. And we also wanted to assess the potential impact of stocking history that it could have had on the white brown trout population. Then, depending on our catchments that we were studying at the time, we might have just a few specific objectives to that catchment. For example, the Loch project, we also did a lot of work on the ferret trout. And in our Moy project, we will have the sea trout elements as well. So we look at where we have done our genetic projects to date. And the map and the table will show you where we've been. The list of catchments we've been is in order when we did them. The very first one that we started on was Loch. This came about through a PhD study, and that provided an opportunity to look at the genetic structure of trout from the carb catchments. And that project really sparked our interest in genetics, because we could see the information that we were able to provide. It also we started asking a lot more questions. So that led then to a whole suite of other catchments that we've done. The Loch was our first one, but we've actually done it a second time as well. Then we've also Mask and Enel, Boyne and the Shore, the whole suite of them there. One leads on to another. Our more recent one, we're about to actually embark on, we've done a little bit of background stuff on it, is Loch Melvin, so we're really excited about that. As we know, we have our ferret skills on it and lots of barriers, so we're very interested to see what the outcomes of that will be, but that's the one we're just about to embark on. So case studies, I'm just going to give some of the bullet point headings or some of the results that we got, and I'm going to talk about Loch and Loch Shooting. I'm not going to mention Delanty at this time. We've run out of time, so we've had to scratch that for the moment, but there are reports available. So Loch brown trout genetics. This is the front cover of the report, and the reports are on our website. So for anyone that's looking for more detail, because this is not going to give a huge amount of detail, you can find our reports and you can download them and read through them. Okay, Loch catchment. And this shows our sampling. The green are our river juvenile samples, and the red are our adult lake samples. Now, because we moved to scales, taking scale samples for DNA extraction, we actually had the opportunity to bring in some of our historic samples. So, which was brilliant, because we've had scale samples from the 80s and the 90s, which allowed a bit of a comparison and to see how things might have changed in that 20-odd year period. So this map has a combination of all of those. And in total, we had 926 juveniles, 710 adults. So a good sample size. Okay, I'm not really going to get into the genetic results, that's in the report, but just 1 or 2 points about it. Geneticists use a number of computer programs for their analysis, and one of them is called structure. So the first stage of the structure analysis, which carb genetics indicated the presence of 2 distinct genetic clusters. This was the Loch East and the Loch West and North. And that grouped all the rivers on the east into one cluster, and it grouped all the rivers on the west and the north into another cluster, which actually tied in really well with the geology of carb, because it's split in the same kind of east-west. The west being more granite and east more limestone. So that was the first level. And then I'm not going to go through all this in detail, I'm just going to focus on the bottom line. But what the structure program does is it takes those 2 clusters, and it looks at breaking them down again into further population. It keeps doing that until it can find no further population substructure. And that's the bottom line here that you can see. So this is shown, and that little white line shows the breakdown each different genetic population. So each of the systems that we sampled in the carb really did come down to a unique population. But I will say that while all the rivers were genetically distinct, some of them showed a high degree of genetic similarity to each other, so they were grouped. So instead of having, I think there might have been 17 or 18 river systems on the previous slide, we actually, for that Corrib, the data generated in that study was best explained by 8 major genetic groups. So we have grouped some of the rivers because they were genetically similar enough to do so. But in all cases where we did group them, it was to a nearest neighbour, so the river may be right next door to it. And from the 8 genetic groups that we've come up with for Corrib, you can see here that these first 3, these are all actually from the Clare system. So within the Clare system, we actually had a number of different baseline populations. So even within one river, you can have quite a number of different populations. And then when you look at number 4 here, it's 3 different rivers. They all came in as one. So they've been grouped as one. The Cong River and the Cong Canal, which makes sense. They're, as I said, they're beside each other. Portimone and Glen Black, Young Riff, and the Drameen and the Harbour. So using the juvenile baseline population, we looked at the Corrib adult assignments. And here we're using the 2012 fish stock survey that we did. We had 393 adult fish. And you can see here in red that 26.9% of those adult fish assigned to the Aboriginal Grange. That is where they spawned. And the table shows the rest of them as well. That's quite a lot from the Aboriginal Grange. It's a big producer, big contributor of the adult stock. Now, one caveat I would say is this is based on 2012. The percentages could vary a little bit depending if we went back there now or 20 years time. It's a good indication that the person is actually quite a significant contributor to the adult stock. The other thing I'd like to point out from this table are the last 2. So you can see that the farm actually, none of the adults we sampled in Corrib actually assigned to the fish farm genetic structure. There's no sign of that at all. It's great because there's a lot of stocking in the 60s and the 70s in that Corrib. And then the unassigned really is trip to uncertainty. So geneticists uses p-value of 0.7 or higher, and if the fish doesn't get that value, then you don't feel the certainty is there to say that came from a particular tributary. We put them in the unassigned. And this unassigned can actually maybe be caused by perhaps maybe straying fish going from one river to another and then spawning there. It can also mean that we have a number of baseline populations that we didn't sample. And that's why we have 35% unassigned in Corrib, because there were a number of very small tributaries and 1 or 2 larger catchments that we didn't sample. And that's possibly reflected in the unassigned. One of the other interesting things on our Corrib was we had GPS for all the adults, so we knew where they were in the lake. And when we plotted them out, and I just showed these 3 examples here, example the Abbott River. So fish that came from the Abbott River down into the lake, this is where we found them. You can see the majority of them are still here in the lower lake. Only a small number of them actually migrated up to Upper Corrib. And then the reverse is seen when we look at Upper Corrib. Beyond the Blackcorn and Mona rivers coming in here in the northwest, quite a few of them along the shoreline and quite a few of them here in the mid regions, but only a very small number of them migrated down to Lower Corrib. On the third map here, you have the Unriff River, and the adults that we sampled in the lake, assigned to the Unriff, were mostly found here in the middle, very close to where the Unriff comes into that Corrib. But again, very few found in Lower Corrib. So there's not an awful lot of mixing between Upper Corrib and Lower Corrib. But as usual, you'll always get a small number that do travel large distances. We have seen this in a number of our other studies as well. We're going to move on to the Loch Sheelan. Again, this is the front cover of the report, and it's on the web to download if anyone's interested in more details. Here we have the map showing the tributaries that we sampled. And the pie charts are actually the genetic structure, which I'm not going to explain here. It's the report if you need it. And you can see that data generated for Loch Sheelan suggested 7 baseline populations. It's interesting to note here that Carrick stream, which is this little one over here, actually had two baseline populations. And then with the Maher Crover stream, which is over here, actually had two as well. And then we had the Upper Inny and the Mount Regions. Lower River is not considered one, it doesn't flow directly into Lough Sheelan, so that's why it's kind of highlighted in yellow. And then the Ross Gray farm elements, that doesn't flow into Lough Sheelan. So when we look at the Adams assignment, we can see that again here we've grouped near neighbours because of the genetic similarity. But we can see that the Mount Nugent and the Crover system contribute 60% to the adult population. That's quite a lot. But when you consider the size of the Mount Nugent catchment, it's the biggest. It's the biggest in Lough Sheelan. So that's really not a huge surprise. I mean, the Carrick and the Maher are quite small. And then you had the Upper Inny, which was the second biggest, and it contributes 13.8%. The others are much lower. I mean, they're really incidental, really. But what we did find is that 18% of the adults actually did, they were Ross Gray fish farm, they assigned to the genetic structure there. But again, that's not surprising because up to that time, Lough Sheelan was annually stocked with brown trout from Ross Gray. So we did catch farm fish in our samples, and we can see that in the results here. But more importantly, though, it was baseline populations didn't have any huge impact or much signals from the Ross Gray genetic structure. So they're not there in the baseline populations. We're only finding the adults that were actually stocked out. The other find here for Lough Sheelan was only 6.3%. This is probably, again, possibly due to a bit of strain, but also there was only one tributary that we didn't actually sample in the Lough Sheelan catchment. So, you know, that's a reflection of our low unassigned value here. Most of the fish came from the Mount Region anyway, so there wasn't a huge amount of strain noted for this catchment. Then I'm just going to maybe give an overview of the summary findings, and this is based on all of our studies so far. What we're saying is that there's a very high level of brown trout genetic diversity throughout all the catchments studied. There was virtually no impact from stocking of hatchery reared fish. All the studies showed no major evidence that would suggest substantial introgression of hatchery-derived genes into the wild. All of our lakes and large main channels have a mixed stock of brown trout. That is, they have a contribution from a number of different sub-catchments, which is great. Genetics can also provide information on gene flow through a system, which means fish moving from stream A into stream B, or is it from stream B into stream A? You know, we can get that information. Some catchments have a high gene flow, some catchments have a low gene flow. Genetics can also identify unique groups within a system that may warrant for special protection, such as maybe above beach barriers, waterfalls, or in the case of the carb or ferrous trout. So you can see that these are, while everything, all our tributaries were unique, some of them are even more unique and quite different, and need maybe a lot more protection, because if anything happened to them, we may not be in a position to get them back. And we were able to say that most populations in all the system studies so far can mainly be explained by the presence of current and or historical barriers, water quality issues, or habitat diversity. Okay, I'm going to finish up on this slide, the fishery biologist toolbox. So we know that before making any decisions on how to manage a catchment, we need to look at a number of elements, and we do our normal baseline surveys, physical and ecological surveys. We look at fish stocks and electrofishing. We look at water quality, the habitat, and all those elements are important when it comes to the management of wild brown trout stocks. But now, through these programs, we can see the importance of genetics, and we definitely feel that it warrants a place in the fisheries biologist toolbox. And if while we have a lot of background information now, if there's the opportunity to expand this, then we should probably use that opportunity. Okay, that is it. Thank you all for listening, and I'm going to hand you back to Lorraine.
Thanks, Karen. And unfortunately, the sound was really bad. And I would say we may have to call you back, Karen, to do this again in the winter time.
Oh, I'm sorry.
But a few people, we got most of it, I think. I could hear it okay, but a few comments came in, or questions came in about the sound.
Okay.
So anyway, I've just got a couple of questions for you. Are there any genetic studies done on Lough trout populations?
Yes, Lough was one of our projects, and we are still writing that one up at the moment. Lough is a really big system. We extended it into the River Sug and the Eddy system, and we took in a number of lakes. So it's a much more complex one, and we're working with Queen's to finalise that report.
Okay. Next question is, were you able to link the physical characteristics, for example, the spottiness, to specific genetic populations on rivers?
No, we didn't. We didn't actually ask the geneticists to tease that part out. It was just one of those things that sparked our interest and led us to ask questions about genetics, but we didn't actually look at that particular gene in the structure to see was that something. And again, it's one of the questions that maybe we could ask, but for us, we were more important at this stage in just identifying what the trout structure in Ireland was like and our subcatchments. And the next levels will be specific questions, maybe like one of those.
Okay, next one is, can you give us a hard example of where the data has been used to protect unique brown trout populations?
Well, the ferrets in Carb is probably the best one. And while staff and anglers alike would have known that the ferrets were using the Carb or the Con River and the Con Canal, it's really good to have the scientific evidence to prove that. And now that we do, because we can see that the Con River actually is more important to the Carb, where the Con Canal is more important to Lough Mask. So if something happened, maybe a fish kill or some incident that wiped out the population there, it's how would we get that back? So there is a potential maybe looking at the Con Canal to maybe repopulate or vice versa, but they were still genetically distinct. So you might have to put in tighter measures to ensure that angling is more regulated, or water quality is kept a tight eye on that, or anything that, like planning developments, that nothing going on in that system that might impact on water quality and the habitat, because then the knock-on effect to the barrack trout, because this is I won't say it's their only spawning, but nearly 99.99% of the spawning is in this little river. We also had a number of other little samples, and the reports probably must show that in the Carb again on the Clare system, the very upper Clare, the Flasca, had a very unique genetic profile that the geneticists said they didn't see anywhere else. Again, this was really probably it's up in the upper system, and we'd have to consider what would happen if that population got wiped out due to habitat or due to water quality. So we'd have to be very careful about what we allow happen there. So it's part of the management. If we know where there are very sensitive, unique populations, we have to be careful about any planning that comes in, or any changes in water quality, or anything, that list of anthropogenic pressures that we went through, if they're going to be impacting. And it is quite useful to maybe repeat some of these studies in 15 to 20 years to see, has there been any change.
Okay. Has any Corrie Lake trout featured in any of the studies, thinking of Lough Bray or Lough Tay or Lough Dan and Wicklow?
We haven't done anything on them, no. And I don't know if there's been maybe smaller projects with the universities specifically themselves, not that I recall, but we haven't done them, no.
I'll just do a few quick ones, Karen, if you can answer quickly. Any information on gathering migrating brown trout to sea?
The Moy project, again, that report is in progress. It's not complete yet. That is going to have a sea trout element, and there's a few interesting things coming up about the sea trout in the Moy in particular. So maybe when that report's out, it might answer some of those questions.
The genetic survey on the North catchment, is that information available?
We haven't done a genetic survey on the Lower.
No.
I don't know, maybe a university or some other project has, but we ourselves haven't done it, no.
And the Lough genetic survey, is that available?
That started as a PhD, so and it was funded by the EPA, so they possibly would have some information. I'm not sure if we can get access to the PhD. And we weren't the lead partner in that, so we were just contributing samples and some background information. But I think Queen's do intend to do another report that focuses on the whole catchment. The PhD obviously would go into a lot more detail. I don't have any exact paper or link to any publications that came out of that PhD, but the PhD would be there. We did present some information on the Lough genetics at our conference a number of years ago, and that presentation is available on our website. While the information won't have changed, it'll give people an insight to what the findings were.
For the both of the lake surveys, did you have genetic samples from the farmed or the stock fish? Is that what you used to assign the farmed origin?
Yeah, so we took samples from Roscrea. We would take the adults and we would take juveniles, and Queen's University have done all this work, so they have that on file more or less. So they would use that genetic profile to look. We automatically do it now on every catchment because quite a lot of our brown trout catchments had stocking historically. So they would always run that genetic profile against wild trout or whatever catchment we're in. So it came directly from Roscrea.
A question, just now that you have the baseline for rivers in the catchments, do you plan to take more adult trout samples to compare?
Because we're able to use scales now, we automatically take scales from every lake survey we do or our electrician. So we have them permanently there. And that's why, as I was saying, that we're able to use the historical ones because we're able to use the scales. So while we're not necessarily planning on repeat genetic projects in some catchments, we actually do have scales. Every time we go there and take do a lake survey or a large river survey, we will be taking those scales. And if an opportunity comes in 5 years or 10 years to rerun the program, then we can use those scales because once they're stored properly and dried out, then the scale sample will be usable.